We're diving deep into the world of immunohistochemistry with a focus on the Inhibin test. This test plays a crucial role in diagnosing certain ovarian and testicular tumors. So, if you're a medical professional, a student, or someone interested in pathology, this video is for you. Let's get started. Pre-test information. Before we begin, let's talk about some important pre-test information. For the Inhibin test, it's essential to provide a copy of the histopathology report. This report gives a detailed analysis of the tissue biopsy, which is critical for the pathologist to interpret the immunohistochemical results accurately. Additionally, the site of the biopsy and a thorough clinical history are required. The clinical history includes patient symptoms, medical history, and any previous diagnosis. This comprehensive information helps in understanding the context of the test results. Specifications of the Inhibin test. Now let's discuss the specifications of the Inhibin test. This test is specifically designed to detect the presence of Inhibin, a protein hormone produced by the Granulosa cells of the ovaries and Sertoli cells of the testes. The test is primarily used to differentiate between sex cord stromal tumors and other types of ovarian neoplasms. Inhibin immunostaining is also helpful in identifying potential steroid hormone-secreting cells in the non-neoplastic stromal component of epithelial cells, germ cells, and other ovarian tumors. Diseases associated with Inhibin test. Next, let's talk about the diseases associated with the Inhibin test. This test is particularly useful in diagnosing sex cord stromal tumors of the ovary and testis. These tumors include Granulosa cell tumors, Sertoli cell tumors. These tumors are often positive for Inhibin, which helps in differentiating them from other primary ovarian tumors. Specimen used in the test. The specimen used for the Inhibin test is typically a tissue biopsy. This biopsy is taken from the suspected tumor site, which is then processed and analyzed in the pathology lab. The tissue is subjected to immunohistochemical staining to detect the presence of Inhibin. Components included in the Inhibin test. Let's move on to the components included in the Inhibin test. The test involves antigen retrieval to unmask the epitopes and formalin-fixed tissue samples, primary antibodies specific to Inhibin, secondary antibodies that bind to the primary antibodies and are tagged with a detectable marker, usually an enzyme or a fluorescent dye, chromogen to visualize the antibody binding, resulting in a color change on the tissue section. Test report time. One of the common questions we get is about the test report time. The Inhibin test typically takes around 7 days to complete. This includes the time required for tissue processing, staining, and analysis by a pathologist. General test information. Finally, let's discuss some general information about the Inhibin test. Inhibin is a useful marker in the diagnosis of sex cord stromal tumors of the ovary and testis. It is particularly valuable because it helps to distinguish these tumors from other primary ovarian neoplasms. Remember, only Granulosa cell tumors and Sertoli cell tumors are typically positive for Inhibin. Inhibin immunostaining also aids in identifying potential steroid hormone-secreting cells within the non-neoplastic stromal component of epithelial cells, germ cells, and other ovarian tumors. This makes it an indispensable tool in the arsenal of diagnostic pathology.